Let's consider the overall process of urine formation. The nephron is the unit of kidney function. There are about 1 million nephrons in each kidney, and each nephron has the ability to make some amount of urine. So let's take a closer look at the nephron. Here is the glomerular capsule, opening into the PCT, or the proximal convoluted tubule. Here's the nephron loop, opening into the distal convoluted tubule. And finally, we have the collecting duct. You should be able to draw this on your own and label it. Here in front, we have the glomerulus, the afferent arteriole, and efferent arteriole. Molecules move from the glomerulus into the glomerular capsule. This is the first stage of urine formation. We call it filtration because molecules are filtered through the glomerulus. The next stage of urine formation is the movement of molecules from the PCT to the peritubular capillaries. This is called tubular reabsorption. Tubular reabsorption occurs in many parts of the nephron, but majority of it takes place in the PCT. The next stage of urine formation is getting rid of molecules that the body does not need. They could be either toxins or they could be ions for ionic balance. It's removal of certain ions that can bring about acid-base balance. So this is the removal of unwanted molecules. So that's called tubular secretion, and that happens in the DCT. The final stage of urine formation is water conservation. Depending upon the needs of the body, the water needs to be conserved or lost. And that is mostly happens in the collecting duct, and we call this stage water conservation. And whatever comes out of this process is that little drop of urine that you see. Filtration is the first stage in urine formation. This takes place in the glomerulus. The glomerulus acts as a filter, and the glomerular capsule is what catches the filter. Here we see the PCT. The glomerulus gets its blood from the afferent arteriole, and the blood leaves the glomerulus via the efferent arteriole. Small substances can pass through the glomerulus and end up in the glomerular capsule and the PCT beyond. Larger substances, such as formed elements and plasma proteins, cannot pass through the filter, and so they end up in our general blood circulation. If these large components of the blood end up in our urine, then this means that the filter is broken. Now, the substances that can pass through the filter are glucose, amino acids, ions such as sodium, potassium, calcium, chloride, water, and urea. You want to imagine the filter to be like a tea filter. The big tea leaves stay in your filter, and those would be the formed elements and plasma proteins. And the small tea leaves that actually pass through the filter and end up in your cup are the glucose, amino acids, ions, water, and urea. Here we have the main difference here is what can and cannot pass through the filter. That's the main thing. Tubular reabsorption is the second stage of urine formation. This happens in the renal tubule. The renal tubule is made up of the proximal convoluted tubule, or the PCT, the nephron loop, and the distal convoluted tubule, or the DCT. Majority of tubular reabsorption, however, occurs in the PCT. It is the movement of molecules from the PCT to the capillary here being the peritubular capillary that comes out of the efferent arteriole. So here is the movement of molecules such as glucose, ions, amino acids, and water move from the PCT to the capillary. Now, in order for this to happen, these molecules must actually travel from 1 to 2 to 3 to 4. 1 is the lumen, 2 is the tubular cell, 3 is the interstitial fluid, and 4 is the capillary. So let's write those things down. 1, lumen, 2, tubular cell, 3 is the interstitial fluid, and 4 is the capillary. So molecules must enter the tubular cell, leave the tubular cell, and enter the capillary. So let's look at the movement of molecules from the lumen of the tubule into the tubular cell. So we are looking at molecules such as sodium, potassium, chloride, glucose, amino acids, all moving from the tubular lumen into the tubular cell. If you remember, the lumen is the space in the tubule. So all of these molecules, they move into the tubular cell via secondary active transport. If you remember, secondary active transport is powered by the movement of sodium down the concentration gradient. And glucose, amino acids, and other ions can catch a free ride with sodium into the tubular cell. So all of them enter with the help of sodium. Now, what happens to the water? The water always follows the salt. So it is obvious that the water will follow the salts into the tubular cell via osmosis. So let's look at the movement of molecules from the tubular cell to the interstitial fluid. So molecules would have to leave the tubular cell in order to enter the interstitial fluid. So the molecules that we are talking about are again sodium, potassium, chloride, glucose, and amino acids. Sodium, as we remember from the secondary active transport that we studied earlier, sodium enters the cell via secondary active transport, but it has to be pumped out of the cell, the back end of the cell. And so here we see sodium leaving the back end of the tubular cell via primary active transport. All other solutes will leave the tubular cell via facilitated diffusion, which means each of these solutes will have their own channel. So there will be a glucose channel, potassium channel, and everything else. Water follows via osmosis. Now the molecules that are in the interstitial fluid must enter the capillary. We must remember now that the capillaries in the kidneys are fenestrated capillaries, which means that they have holes in them. This allows the molecules that are in the interstitial fluid to enter fairly easily and quickly into the capillary. So we have a fenestrated capillary that is number 4. So the molecules, all the solutes, sodium, potassium, chlorides, ions, glucose, amino acids, all of them enter the capillary via simple diffusion down their concentration gradient. Water, as always, follows the salt. So since all the solutes are in the capillary, water easily moves into the capillary via osmosis. The third stage of urine formation is tubular secretion. This is the movement of molecules from the capillaries to the renal tubule. Whatever ends up in the renal tubule will become urine. There is no more absorption beyond this point. So let's take a look at the capillary, which is the peritubular capillary. This is also a fenestrated capillary. The part of the renal tubule that does most of the tubular secretion is the DCT. So let's imagine that this renal tubule section is the DCT, and molecules move from the peritubular capillary to the DCT in the opposite direction as we've seen before. 4 is the peritubular capillary, 3 is the interstitial fluid, 2 is the tubular cell, and 1 is the lumen. So we're going backwards, 4, 3, 2, 1. The molecules that get secreted are potassium, hydrogen ions, and some toxins. Let's consider the secretion of potassium from the capillaries to the renal tubule. It is important to get rid of potassium from the blood because too much potassium in the blood can cause cardiac arrest. So we have to go backwards. 4 is the capillary, so the potassium must leave the capillary, and 3 is the interstitial fluid. Now the potassium must enter the tubular cell, and then it must leave the tubular cell to enter the lumen. So we have potassium leaves, potassium enters the tubular cell, and potassium leaves the tubular cell. Potassium leaves the capillary via simple diffusion because the capillary is fenestrated, so it's easy for the potassium to leave. Potassium enters the tubular cell via primary active transport. If you remember, sodium was pumped out of the tubular cell via primary active transport. There's a sodium-potassium pump here, so the potassium is pumped into the tubular cell. Then potassium leaves the tubular cell via facilitated diffusion to enter the lumen. Once it enters the lumen, it leaves out as urine. Let's consider the secretion of hydrogen ions. If there are too many hydrogen ions in the blood becomes too acidic, and a condition known as acidosis results. The hydrogen ions ultimately come from too much carbon dioxide in the blood. So when carbon dioxide mixes with water, it becomes an acid, and that acid releases a large amount of hydrogen ions. So here's the carbon dioxide leaving the capillary fairly easily, diffusing out of it, and diffusing into the tubular cell. Once inside the tubular cell, the carbon dioxide mixes with water and becomes carbonic acid. The carbonic acid then dissociates into hydrogen ions and bicarbonate ions. If the carbonic acid levels increase, that's the carbonic acid H2CO3, when that increases, the level of hydrogen ions also increases. And the hydrogen ion levels increase within the tubular cell. So there you can see the hydrogen ions increasing. So this hydrogen ion is pumped out of the tubular cell via primary active transport. And once it leaves the tubular cell and reaches the lumen, the hydrogen ions leave the body along with the urine. In this way, the kidneys are able to achieve acid-base balance. The last stage in urine formation is water conservation, and this happens in the collecting ducts of the nephron and the DCT. On a hot day, the posterior pituitary releases ADH, the antidiuretic hormone. Antidiuretic hormone makes the collecting duct retain water. In other words, the collecting duct becomes permeable to water, as does the DCT. So the water enters the capillaries and the water is retained, so the urine that comes out is more concentrated. Alternatively, on a cold day, the posterior pituitary does not release as much ADH. So the collecting duct and the DCT are less permeable to water. As a result, less water enters the capillary, and so the urine that is produced is more dilute.